everyone. We're going to continue our discussion of cellular respiration by discussing the first pathway, or glycolysis. Okay? So glycolysis occurs in the cytosol for both prokaryotic and eukaryotic cells. There are some differences of where certain parts of this process of cellular respiration occur between prokaryotic and eukaryotic cells, and we haven't really discussed them yet. But glycolysis occurs in the cytosol for both prokaryotes and eukaryotes, so we're starting here. Okay? What are the products of this reaction? Well, ATP is produced, NADH is produced, and then also pyruvate and water molecules are produced. Okay? All right, let's start with the details, or let's get to the details. Glycolysis begins with one glucose molecule. Okay? This one glucose molecule is ultimately transformed into two pyruvate molecules plus two water molecules. All right, pretty simple. Glucose, glycolysis, the splitting of sugar. So here is a glucose molecule. It is a simple sugar, and it's a 6-carbon sugar. And this 6-carbon sugar is actually split into two 3-carbon sugars, which then ultimately go on to are transformed through the use of enzymes into two pyruvate molecules, which I drew a pyruvate molecule here, which is a 3-carbon molecule. Okay? So glycolysis means literally the splitting of sugar. Well, how does that happen? Well, glucose is first transformed through a series of steps using enzymes, and we talked about enzymes in the last chapter. You did your enzyme lab. You realized the importance of enzyme in all of these chemical reactions. So in the first part of this pathway, glucose is transformed into glyceraldehyde 3-phosphate. Now, in order to do that, the cell must use two ATP molecules. So there's an energy investment phase of this pathway, okay? But after you get to the two molecules, actually, of glyceraldehyde 3-phosphate, other enzymes are then used to transform these two molecules into the 2 pyruvate plus 2 water molecules. In this process is where the payoff really is obtained. The cell obtains 2 NADH molecules plus 2 hydrogen ions. Do not lose track of those hydrogen ions. They are important when we get to oxidative phosphorylation. The cell also gains 2 ATP molecules, transforms 2 ADP into 2 ATP, and it does it again in this half of the pathway. 2 ADP are transformed into 2 ATP molecules. So you could say there's two halves of this pathway: the energy investment phase and then the energy payoff phase. Okay? Down here, if oxygen is present, these pyruvate molecules will go on and enter the mitochondria through active transport and enter the citric acid cycle. If oxygen is not present, then the pyruvate molecules will go on to be used in the process, another catabolic process called fermentation, and we'll talk about that later, okay? So the question you're probably asking yourselves right now, especially since you've read your textbook, is, do I really need to know every single one of these steps and all 10 enzymes? There are 10 enzymes involved in transforming glucose into the pyruvate plus water. And my answer would be no, okay? And I hope we're right on this. But do you need to know every single enzyme and every single step, or every single intermediate product? I would say no. But there are ones that are extremely important. First of all, you need to know like the reactant and the product. That we start with glucose and we end up with 2 pyruvate plus 2 water molecules. That's essential, okay? If you draw out this pathway showing the 10 enzymes like this and just boxes for the intermediate products, I would say that there's some enzymes or some points that you really need to know. I would say you need to know this product right here, which is the glyceraldehyde 3-phosphate, okay? How many molecules of these are produced? Well, one, technically. So glucose is split, or this product, intermediate product, is actually split right at this step by this enzyme right there, which is called aldolase. And it's split into these two products. It is the glyceraldehyde 3-phosphate. It's only this product that continue on in the glycolysis pathway. So what's going on here? Well, these two isomers are at equilibrium. However, not at equilibrium, sorry. They never obtain equilibrium because this product's always removed. So this reaction is always driven this way. So we actually can ignore this intermediate product, I would, entirely. So aldolase is important because this is where the glycolysis pathway gets its name. This is where a 6-carbon sugar is actually split. This is the reaction where 2 3-carbon sugars are formed, okay? Then eventually, the glyceraldehyde 3-phosphate is converted through a whole other series of enzymes to the pyruvate plus the hydrogen, the water molecules, okay? Notice here is the energy investment phase. 2 ATP molecules had to be used to get from glucose to the glyceraldehyde 3-phosphate. This is the energy payoff phase. Notice we get 2 ATP molecules there, 2 ATP molecules there, for a total of 4 ATP molecules there. But we have to remember to subtract the 2 from this phase for a net gain of 2 ATP molecules in the whole pathway of glycolysis. The last thing I would remember if I were you is this enzyme right here. This enzyme is called phosphofructokinase. And why would I remember that enzyme, or why do I think it's important? Because this is the enzyme that regulates the whole pathway, or where regulation occurs. ATP will allosterically inhibit or regulate that enzyme. If there's a lot of ATP in the cell is not going to spend energy, especially here using ATP molecules, to make more ATP. So ATP will bind, or ATP products will bind to that enzyme and shut it down. And so the whole pathway will be shut down from that step forward. So no more pyruvate will be made, and no more water. No more ATP will be made at this end, and no more NADH or hydrogen ions will be made there. Okay? So I would remember glucose, and then the products, pyruvate and water molecules. I would remember the 2 enzymes, one at step 3, the phosphofructokinase, the other at step 4, the aldolase, that gives this reaction its name. And I would remember the glyceraldehyde 3-phosphate right there, because that's the one intermediate product that does then continue in this energy payoff phase of the pathway. So in summary, glycolysis gives us, what is our net gain? We gain 2 pyruvate molecules plus 2 water molecules. We gain 2 NADH molecules, which are extremely important when we talk about electron transfer. You know, moving electrons from one molecule to another without the loss. NADH does not allow electrons to lose a lot of potential energy, so that we can use that energy later when the electrons are transferred through the electron transport chain when we get to oxidative phosphorylation. So NADH is an extremely important molecule, plus the 2 hydrogen ions, also important. Plus, we generate 2 ATP, or the cell generates 2 ATP molecules in this pathway. So that's glycolysis, and what I think are the most important parts of this pathway. And now we're going to go on and discuss, if oxygen is present, what happens once the pyruvate molecules are actively transported into the mitochondria, which is the citric acid cycle.